Okay, good. So, welcome to the third unit. We are almost in the middle. Uh, After unit 5, we will have midterm examination. So we are warming up a little bit. So we are already getting into the real physiology. So today we will have one of the most important metabolic pathways, which is fatty acid biosynthesis. And as usual, I'm going to emphasize what is important to know and to memorize. To begin, cell membrane. Cell membrane is the next component that we are going to cover in this class, simply because we talked a lot about cell wall and periplasmic space and everything what's happening in periplasmic space. We are going to talk more about periplasmic space, especially when we cover in Unit 4 protein transport, secretion, and translocation. But for now, let's just stay in the cell membrane area. So you probably already know that cell membrane is something that separates, it's a compartment that separates cytoplasm from cell wall, so we can call it a cell. And this is the current structure that the cell membrane, that all components that we have in cell membrane. And cell membrane is a very important place where cellular energy is generated, or as it's known as proton motive force or respiration components of the cell. Cell membrane is not only a place for energy generation, it's also a barrier toward the external world, which means it has selective permeability for ions and gases and other molecules. And it's also the place where nutrient exchange is happening. As you probably remember from last week, cell membrane is place where chemotaxis molecules are located, which are molecules that a prokaryotic cell is using to sense the environment. And with all of these roles, we usually say that cell membrane is a critical component that defines all life forms, so and us and bacteria and archaea. And this basically through this cell membrane, signals that are coming from outside world are transferred to the cell, and that's where all that communication is happening. So it's literally like a main role. And again, it's not that cell membrane using cell membrane to communicate with the external world. Also, cell membrane is communicating with its own internal world, which is a chromosome. So it controls chromosome through cell membrane and proteins that are anchored cell membrane to chromosomes. So based on the membrane structure, we do have some genome segregations, which means that cell membrane, even though it's universal for all life forms, it doesn't really have universal structure. It's different between archaea and bacteria and us. Well, actually, we are way more similar with bacteria in terms of cell. So this is the basically, in terms of cell membrane, bacteria and eukaryotes are same. I don't want to say similar, they're same. They have phospholipid structure, but archaea are quite different. So the basic phospholipid structure that we see, LPL stands for phospholipid, so that is same in prokaryotes like bacteria and us. And the main component that makes this cell membrane is glycerol. Glycerol is nothing else but 3-hydroxyl alcohol that can also be used as a carbon source for some bacteria, which we will cover later. The next component of plasma membrane in bacteria and eukaryotes are fatty acids, or Fa, and they're usually attached or esterified to the glycerol moiety, which you can see here in the picture. So this tic-tac structure, that's fatty acid, and they're attached to the two hydroxyl groups of glycerol. The third one is the phosphate group, which is attached to the third hydroxyl group of glycerol moiety, and that's why we call them phospholipids, because they have phosphate and they have fatty acid, so they feel glycerol. The length of these fatty acids again, I'm saying bacteria and eukaryotes because archaea have different structure, but the length is variable in between 14 to 20 carbon molecules. Usually bacteria comes to have 16 or 18, but this actually is not really fixed state. So the composition of fatty acids is changing, and it's very responsive to the temperature and the environmental conditions. And as we will see later, it's responsible to the carbon source that we are giving to molecules. Very often that structure is actually indicating the changes in physiology or microbial cells. So not only in length, those fatty acids can differ into the chemical structure itself. So they can be saturated if they have only one carbon bond, like CC. If they have double bond between Cs, and there can be multiple, more than one double bond, then they're called unsaturated fatty acids. And again, this will vary depending on the environmental context. So, phospholipids are not the only part of the membrane. The next important component are proteins. So while phospholipids, we say that have polar or hydrophilic heads, which is a phosphate, and some other compounds, which we will cover later. And there are also non-polar hydrophobic tails, which are your fatty acids. So literally PL or phospholipid stands for glycerol, fatty acid, and phosphate. So glycerol is the first compound that you need. And without that, you can't build phospholipids. Next compound will be fatty acids that will be esterified. And there are two of them that will be esterified to this glycerol, to this OH. So if they have double bond, they are unsaturated, that will turn into subject to some chemical reaction. There is a saturated fatty acid that are without carbon bond, and this is hydrophobic component. Those fatty acids are hydrophobic. And last but not least is a phosphate, which is usually esterified to the third hydroxyl group, which builds phosphate. Now, I don't know what I pressed. Okay, sorry, my bad. So, how are phospholipids made? So now when we know what are components like glycerol, fatty acids, and phosphate, let's see how they are made. So the key intermediate, of course, to build those phosphate is phosphoric acid. That's what we have to have. Logically, we will need to have glycerin. So these are first two components that you need to have to build phosphate. And of course, you need to have a fatty acid, and they will be esterified to the first two hydroxyl groups of this glycerol, which has 3, and the third one will be the group in place of bacteria will be glycerol 3. Now, further synthesis involves the attachment of some other now hydrophilic compounds, which are usually called R1 and R2. Sorry, they're not hydrophilic, they're hydrophobic compounds. So, bacteria, so we have still talking about fatty acids. Fatty acids can have 16 or 18, or it can have depending on the type of the bacteria. Now, next question is, where is the glycerol going to come from? So glycerol for phospholipid biosynthesis can be made internally, which is de novo synthesis, but there is also another pathway where glycerol can be imported through the cell membrane, and both of them we will cover. So if we're talking about internal or de novo production of glycerol for phospholipid biosynthesis, for that, cell needs to have two compounds. It has to have a synthase, which is this GPSA synthase, and that synthase is actually going to complete the reduction of glycolytic molecules, let's call DHAP or dihydroxyacetone phosphate. And we will learn more about this molecule when we cover glycolysis later on in Unit 6. But DHAP, increased accumulation of this molecule, actually shows that your cell is, the bacterial or microbial cell is going through the process of some changes that will require change of the membrane lipids. So if, for example, one of the common stress responses on ethanol is the change in lipid structure, and when cell is facing with a methanol or ethanol, it will increase the amount of this dihydroxyacetone phosphate because it's struggling to change the lipid structure so it can face the given environmental stress. So DHAP is what needs to be reduced by GPSA to make glycerol 3-phosphate. So the characteristic of glycerol 3-phosphate is, of course, phosphate at the end, and third hydroxyl group. So that's your first component that is now waiting to get fatty acids. This is, and notice that in bacteria, this is glycerol 3 phosphate. So, and this is characteristic, this is landmark of bacteria. So you will see later on why that matters. Now, this glycerol 3 phosphate is going to attach fatty acid. And where is fatty acid coming from? Fatty acid is coming from acyl ACP. ACP stands for acyl carrier protein. We will see in the next slide how all this acyl ACP structure is built. But now this is just a brief overview. Fatty acid comes to be esterified to two remaining hydroxyl groups. And the enzyme that transfers those fatty acids are simply called transferases, acyl transferases, and they create something that is called ester linkage between fatty acids and glycerol moiety. And the reason why I'm mentioning this ester linkage is because in archaea there is something completely different, and that's where the evolution or the divergence is happening between bacteria and archaea. So again, that later on, so those fatty acids can vary in the length, but they have be C16, C18 in bacteria, and the result is bacterial phospholipid. So again, this is all when we talk about internally made glycerol, but glycerol also can come from external sources. So if we want to feed bacteria with the glycerol, bacteria has to be equipped with certain enzymes to be able to incorporate that glycerol from the environment. So that's the bacteria growth on glycerol salt, carbon salt. So there is a transporter, intermembrane transporter, that is called GLP-F. So that transporter recognizes glycerol that you externally give to bacteria. And when glycerol comes in upon entrance, it immediately gets phosphorylated by kinase, which is called GLP-K. And the result is glycerol 3 phosphate that later on can be used for further membrane synthesis. Why this matters? Well, it matters because in that way you can actually adjust cellular tolerance on, for example, nowadays that matters for methanol because it can remodel its lipid structures. And this is the structure. This is actually something that me and Nina are currently working on with. We are trying to clone this structure into our bacteria. Doesn't really go so easy. This thing is intermembrane protein, so God help me if that will ever work, but we'll do our best. At least we will clone, right? We will one day. So anyway, the formation of dihydroxyacetone phosphate with glycerol 3 phosphate from DHAP is the only de novo synthesis, and it requires BPS. The one that has GLP, F, and K is actually from external source. In terms of fatty acids, so now we'll go a little more into the biosynthesis of fatty acids. So how you will name the fatty acid, that depends on the number of carbon chain. So the number of carbons in the carbon chain. So if you see this C16 and the two dots, one, that means that this fatty acid has 16 carbon atoms and there is only one unsaturated double bond. Again, I will never ask you to memorize these for these structures. That's ridiculous. But there are various types of fatty acids. There are many branch-chain fatty acids. Then usually bacteria that are dealing with certain stresses, like in lactobacillus, you will see unusual fatty acids like with cyclopropane. So in past, fatty acids were a very good indicator on which microorganism you are dealing with. Nowadays, we have DNA, actually 16S rRNA, so we don't use this one, but they're still very good indicator of environmental conditions and physiological changes. So those specific moieties of fatty acids will actually help bacteria to respond to environmental stresses. Again, alcohol, solvent stress is one of the common responses. Again, not fixed, something that is really changing. So that's why it's not the best molecule to use to identify bacteria because it's changing, but it can definitely help to respond to certain physiological questions like how is cell dealing with a certain level of stress. And we will see that when we come to the unit 5 when we talk about different stressors. especially temperature stresses. So we covered briefly fatty acids. Again, it's not the end. We will talk in details about biosynthesis of fatty acids. A little bit more about polar head groups. So fatty acids belong to the hydrophobic area, like that's hydrophobic tail of phospholipids. Polar head groups, which can be all these guys listed here, phosphatidyl ethanolamine or PE, PSPG, and UPG. Again, knowing abbreviations will be fine. Those are very complicated names. But more important is that those polar head, these are polar components, hydrophilic components, and they actually affect the way how lipid will interact with its environment, because those heads are actually exposed to the internal and to the external environment inside. So in summary, what phospholipids will have: glycerol moiety, phosphorus, esterified, and attached various fat groups into the phosphate. And you have two fatty acid chains, which are with saturated and unsaturated carbon. So this is the basic structure of phospholipids, and we already know where the glycerol is coming from, so we talked about this. And I won't go in details with polar head groups, but fatty acid biosynthesis is something that I will cover right after archaeal phospholipids. And the reason why I'm mentioning archaeal phospholipids, even though we are not going in details about that, is simply because this is where evolutionary difference between bacteria and archaea comes into the point, and us actually. So we and bacteria are on one side with phospholipids, archaea are in different side. And if somebody took my class last year, Bio 322, there is a table that I'm quite insisting in that class for people to know. So the main difference between bacteria and archaea phospholipids is, in bacteria, on glycerol moiety, there is a fatty acid ester bond. So it's ester bond. But in archaea, there is ether bond. And it's not fatty acid in archaea, it's isoprene. So the difference is, in archaea, you have ether, not ester bond, and instead of esterified fatty acids like in bacteria, you have isoprene. And the reason is quite easy to understand. So isoprene has 5 carbon molecules, and isoprene is more complex, and it creates very rigid and strong structure. And the reason why this matters for archaea is because they evolved in very harsh environments, like very extreme environments, and that's why they had to develop this type of lipids so they can deal with high stressors. So that's the only reason why they have this specific ether bond. So archaeal phospholipids are related to rigidity, survival in extreme conditions, and they're actually variable. Again, it will depend from the environment where they are. And the name of archaeal phospholipid is archaeol, the ether lipid, but they also have different moieties attached, R1 and R2. They vary in length. They usually have more carbons than in bacteria, so 5C atom, and there are cyclic rings in archaeol, again, so they can deal with stressful conditions. And archaeal phospholipids are the mode of evolution. Why? Again, that's where diversity comes into the game. So you always require glycerol 3-phosphate. So glycerol still required, phosphate is still required, and it will always, in both cases, actually in all 3 cases, it will come from glycolysis, because that's the so it's still all like that. But the difference is in the enzyme that forms these polar head groups. So in archaea, which are exposed to the extreme environment, the enzyme that creates glycerol moiety makes something called 1P. And what means 1P? That means that the phosphate is on the same side structurally as OH group, as you can see here. In archaea, that's in archaea, in the same phosphate. In bacteria, that bond is split, and you can see that here you have glycerol 3-phosphate. So there is so these guys here are on the same side, OH and phosphate, and on 3-phosphate they are on different sides. That's the difference. That's the mode of evolution. Now, and these main differences between bacteria and here again, I'm not putting eukaryotes here because we are not talking about eukaryotes, but general comparison is in this table, and this is something that I quite insist people to know. So in bacteria, you will see ester linkage between side chains and glycerol phosphate, and you will see fatty acid side chains. In archaea, you will see 1-phosphate, and you will see isoprene side chains. It's ester bond in bacteria, it's ether bond in archaea, and in bacteria you will see C16, C18 common length, while in archaea will be many more carbons, like 20 and 25. So in bacteria you will have temperature sensitivity, while in archaea it's going to be temperature resistant, because that's how we function. So, any questions so far? Yes, Athena. Let me see if I get moving in there.
When you were going over your lifestyle, yes, right? The bacteria for one of the other is not. It's really kind of happen a lot.
It actually doesn't happen all at once because it doesn't happen all at once because you don't always give bacteria glycerol all the time. If you give bacteria glycerol as a carbon source, it will again depend on what it has and what's the metabolism rate. If you have to use, I would say, like, if I have to make everything yes, and that's exactly what I'm doing, because DHA, the hydroxyacetone phosphate is the product of fructose 1,6 phosphate. So you will have DHA and you will have glycerol 3-phosphate. So you can always have glycerol 3-phosphate going down and make more energy, and DHA can go and make phosphate. Now, if you give them external glycerol, again, they have to invest energy to transport it over the membrane, and then you have kind of the quality. But still, you can invest that DHAP to make more energy, so it will prefer the one on the top. So it will really be the balancing between what you give them and how much energy to take, how many NADH, how much ATP you can get. So it's going to be trade-off. So there is no really, the only way how you can trade how bacteria will prefer is to give them glycerol, then it will balance its energy in terms of what's kind of acid. If you don't give them glycerol, they don't really have a choice. Many of them don't even have glycerol transporters, so you would have to add them there. Now then the question is again, how much energy will that transport across other things? Does that answer the question? Okay. Any more questions? Okay, so fatty acid biosynthesis. This is when people usually freak out when they see metabolic pathway. And this is one of the first, although we did have peptidoglycan biosynthesis. So we will go briefly over general overview of fatty acid biosynthesis with this simplified scheme, and then we will dive into the details. So the fatty acid biosynthesis starts with condensation, and it involves acetyl coenzyme A and malonyl coenzyme A. Acetyl coenzyme A, of course, comes from metabolism. That will be way more clear when we talk about bioenergetics and we talk about TCA cycle and actually glycolysis in the TCA cycle. So acetyl coenzyme A comes from carbon metabolism, central carbon metabolism. So ACP, or acyl carrier protein, is the one that carries acetyl coenzyme A molecules together. But it technically connects acetyl coenzyme A and malonyl coenzyme A. But malonyl coenzyme A is built of acetyl coenzyme A through carboxylation. Now, just breathe. So think about acyl carrier protein as something that actually carries stuff to start biosynthesis. So let's stay on acetyl coenzyme A first. So the enzyme that is required for this is going to be 5 things. So all these structure of enzymes is called 5 enzymes. So H is the first one, Fab H, that does condensation. And it's important, you will see later on, because it's antibiotic touch. So this Fab H is actually doing condensation of acetylcoenzyme and malonyl coenzyme. As the chain is growing, there will be some analogs of Fab H that will take over condensations, but initial one is Fab H, and that's the one that you have to know. So condensation is interaction between malonyl coenzyme A and acetylcoenzyme A, and this is done by enzyme Fab H, the first one. This reaction will release one carbon dioxide, and the next step is catalyzed by a reductase. Reductase is called FabG. It's of course doing reduction, and that reduction involves one cofactor. So NADPH is oxidized to NAD, acetoacetyl is reduced, and then the next step is dehydration or dehydratation, which is generating linear fatty acid chain, and this is catalyzed by Fab. The next one, and probably the key point, is Fab I, which is next reduction. It creates formation, it causes final reduction and formation of acyl ACP. So this acyl ACP is actually now carrying basic fatty acid. So this is general overview. So again, in bacteria, depending on what is the number of carbon atoms they have, this whole process can repeat 8 or 9 times if it has 16 or 18 carbon, because it happens in a chunk of 2. So this was general overview. Let's see now from chemical structure how we call all of these intermediates. So again, enzymes are H, G, A, and at the end is I, that does final reduction. So you remember when I told you that acetylcoenzyme A comes from the central carbon metabolism? So that acetylcoenzyme A is converted to malonyl coenzyme A by this carboxylase. It's enough to know that it's carboxylase. And now you have built malonyl coenzyme A. So with malonyl coenzyme A and acetylcoenzyme A, conjugation with Fab H can start. There is this enzyme FabD that malonyl coenzyme A to ACP, because that ACP is important. And condensation enzyme FabH is joining acetylcoenzyme A. Again, acetylcoenzyme A is also connected to ACP, to the malonyl coenzyme A that is also connected to ACP. So ACP is required to carry both of these compounds. The result of condensation is beta-ketoacyl ACP. That's the substrate for next enzyme, so this ketoacyl, and the enzyme is FabG. So this FabG reductase is going to reduce ketoacyl to now hydroxyacyl ACP. Again, we are having it connected to ACP. And this beta-hydroxyacyl ACP is going to be substrate for this dehydratase that is going to result in the structure called trans-2-enoyl ACP. And trans-enoyl will be a substrate for reductase, and it will form acyl ACP. So this is the basic of fatty acid. Now, acyl ACP is going to be used for biosynthesis of phosphorus. And Fab-I homologs, you don't have to know, they are K and L. In different organisms, they're called differently, but the most important for you to know is Fab-I. How is unsaturated bond formed? So you know that we said that we have saturated fatty acid, but now how we introduce unsaturation. So both saturated fatty acid and unsaturated fatty acid can be formed in bacterial cell, and there will be different enzymes that are introduced in unsaturated bond. And we will talk about this more when we cover stress conditions, but when there are non-stress conditions, condensation, reduction, dehydration, and final reduction are occurring normally, like everything goes as we just described now. Carbon-carbon double bond between fatty acid and acyl ACP will be in so-called trans orientation, when everything is normal, like trans and oil bonding. When some stress happens, usually some sort of cold shock, Fab I reduces, activity becomes slower. When activity of Fab I is slower, that facilitates formation of cis bond between fatty acid and acyl coenzyme A. So this is where the cis bond will occur, and cis bond is a substrate for another Fab enzyme that is called Fab B. And Fab B will now introduce double bond, and it will be fixed in the position in fatty acid growing chain. Now, this fatty acid synthesis is a very huge target for antibiotics, and we will cover three of them: triclosan, isoniazid, and platinum-cimicin. And all of them have a similar mechanism, similar like vancomycin and penicillin. So isoniazid, which I used to call isocyanide, and I just couldn't pronounce isoniazid, but it's not cyanide, it's niacid. So this is the structure, and the target of isoniazid and triclosan is the same. It's Fab I enzyme, but the mechanism is different, pretty much like in vancomycin and penicillin. So what's the mechanism for isoniazid? I'm still sometimes forgetting. It's not cyanide, it's niacid. So we talked about condensation that's happening through this Fab HGA enzymes and the final reduction through Fab I. So if you remember, the resulting reduction is giving you trans 2 enol, that is a substrate for Fab I, so Fab I is expecting to reduce. Now, what's happening when you give them isoniazid? So when you give them isoniazid, bacteria has some peroxidase catalase system, which you probably heard about. And with that system, bacteria actually does suicide. So it activates this isoniazid that you gave us. So isoniazid that is activated is forming this acyl radical, isonicotinic acid acyl radical. And that radical, which literally bacteria formed itself, has ability to bind to NADH, and it literally traps that NADH, so it forms this nicotinyl NADH. So why do we need this NADH? And why it matters? So when it binds that NADH, this whole structure, now nicotinyl acyl NAD, binds to Fab I. So bacteria literally made a structure that will inhibit its own enzyme. And it binds to, when this structure binds to Fab I cannot complete final reduction of transenol coenzyme. In past, this isoniazid was used in tuberculosis, Mycobacterium tuberculosis, but the bacterium developed mutation in catalase gene, CatG, so it does not perform, so it literally develops mutation in catalase peroxidase, so it doesn't do suicide, so it learned how to defend. So it cannot bind, so isoniazid doesn't bind to any to us. So that's it. So in resistant organisms, CatG is not able to create acyl radical, and there is no molecule to bind NAD. So as Fab I can finish complete reduction. Triclosan is so the target is also Fab I, but the mechanism is different. So you probably heard about triclosan because it was found in soaps and detergents. Like these are all, and it was forbidden by FDA and the Environmental Protection Agency and the European Union because it creates resistant bacteria. So they just stopped using it. And so how triclosan functions. So again, synthesis is happening through HGA enzymes. So resistance to triclosan is happening with bacterial membrane alterations, but what's the mechanism? Again, Fab-I is a target. So this resulting structure that is substitute for Fab-I, trans-enol ACP, so that's the Fab-I will recognize. So this Fab-I enzyme that is expected to reduce trans-enol coenzyme A has affinity for NADH. Why for NADH? Because NADH has to be oxidized, so that's why it's in reduced form NADH. So it has to be oxidized to NAD because we want trans-enol to be reduced. It's like oxidoreduction. So this is what will happen in a normal structure. So trans-enol coenzyme A, trans-2-enol ACP has affinity for NADH. But when triclosan binds, when triclosan comes in, when it binds to Fab-I, the affinity of Fab-I is changing. So now Fab-I doesn't have affinity for reduced NAD4, which is NADH. It actually has affinity for oxidized form, which is NAD+. And that disables completion of final reduction, so you cannot get fatty acids in. Platency-mycin has a different target. So for where triclosan and isoniazid are have a target technically Fab-I, platency-mycin is targeting Fab-H, which is the enzyme that does condensation. So that's the enzyme that condenses malonyl and acetyl coenzyme A. So platency-mycin, which is antibiotic, I believe, antibiotic from plants, interacts with chiral groups in active site of Fab-H and prevents its further increase. So without that initial step, fatty acid cannot be formed. Some of the interesting fatty acid filters is something that I already mentioned. Again, even though that is not really used anymore in identification of bacterial, like it's not really bacterial ID nowadays when you have 15 srRNA, but microbes do have specific fatty acid profile, and the way how you do this, how you get this profile is you extract all fatty acids from organisms, you esterifize it in reaction with methanol, and you generate something called PAIN, for fatty acid methyl esters. And then you can observe different characteristics of different organisms. So it is very useful to observe how organisms react on different stressful conditions. In commercial applications, this old company, which I don't think it even exists anymore, called MIDI, they were looking fatty acid composition and they were able to distinguish various bioterroristic agents. I don't think it's done anymore. I really didn't check recently because nobody really used this as ID for organisms because it has DNA and 16 RNA. But it's still useful for evolutionary studies and for physiology studies, especially if you're comparing bacteria and archaea. So archaeol has isoprene units that are very stable, so it can be preserved in rocks. So you can extract them from rocks and you can discover which type of archaea was present. So, and now moving on to the cell membrane as a place of respiratory electron transport chain. So we will only start today with the cell membrane. Next class, we're going to talk more about it. It will be basically just 2 or 3 slides just to give you a brief introduction, but I will cover this in the next time when we talk about the transport to cell membrane. So cell membrane is a component of microbial cell that hosts a respiratory electron transport chain, which contains cytochrome oxidases and dehydrogenases, proton complexes, photosystems 1 and 2 if it's photosynthetic membrane. So proton channels, because proton multiforce is extremely important. So now, ATP synthase at the end of electron transport chain, flagellum, secretion system. So those are all components that are located in cell membrane. We will talk a lot about efflux pumps, sensory proteins, solute transports. So we already mentioned flagellum. So cell membrane is a place where all of these events are happening, and There are other molecules that are going to be located, that are going to be associated with cell membranes. So they are hopanoids, which are complex lipids. So they are analog to hopanoids are complex lipids that are also in bacterial cell membrane, and they are similar to eukaryotic cholesterol. So they limit diffusion of hydroxyl radicals. So they have protective role. Then you will have pigments like carotenoids. Important are quinones, which are part of the electron transport chain. So all of these components are powerhouses of the cell, and they are enabling electron transport and proton motive force. Proton motive force is what actually starts all processes in the cell. Bacterial chlorophyll is also hosted in cell membrane. Sterols are big deal, especially in methanotrophs. They have similar structure to our sterols, and they are required to survive oxygen stress in bacteria. So that's why we had this updated fluid mosaic model. So in my time, it was believed that cell membrane is actually made of mostly of lipids. Nowadays, we know that lipids are only 30% of all cell membrane. It's actually 70% proteins. And again, isoprenoid, arterial cell membrane is thicker because it has isoprene compound. So it's quite different, but also more complex cell membrane you will find in phototrophic or chemolithotrophic membrane. Why? Because if bacteria are phototrophic, they have to host photosystems in cell membrane. That's a special type of reaction centers and proteins. Chemolithotrophic membrane, they have to have different equipment in cell membrane because they are using different electron donors and acceptors. So cell membrane is a place where proton motive force is generated. And proton motive force is something that builds the energy for the cell. So because this is, so I will stop here simply because I think it will be too much, and I think we need to digest what we did today. I will stop here. You're good to go. Next class, we're going to start to start on this slide. So let's finish with that membrane is a place where proton motive force is generated, and then tomorrow on Thursday we talk about the ways how proton motive force is generated and two main components of the. Just memorize just Marisa, please write me down. Cool. Excellent.